received numerous awards for his research and has lectured at various hospitals, institutions, and conferences across the country, including here at the CNCF conference several times. He has authored and co-authored so many publications with national and international collaborators, and his presentation today is on CAR T cells for children with neuroblastoma. Dr. Heczey.
Thank you. Thank you for the kind introduction, and I'm really honored to be here. It's really wonderful to get back. Unfortunately, I couldn't join last year. It's great to see some of those of you who I've met in the past couple of years and meet new families. And just wonderful job, Pat. It's great to have us all together. So, as Pat mentioned, my task would be to review some of the work that's been done in CAR T cells. And obviously, the field is really going through an exciting period of growth. So I try to pay attention to some of the nuances and introduce you some of the complexities that come with these CAR T cell therapies, just to give you some ideas that, well, even if the same target is looked for in a certain trial, the cells can be engineered differently. The receptor itself can be engineered differently. So all those complexities can potentially change the efficacy of the product. And that being said, I have some patents related to the technology that are employed, and I was the PI of the GenaK2 study that I will talk about today, but I will do my best to objectively present all the data. So as Dr. Sondal already alluded to, when we say immunotherapy, it's actually a really, really broad basket term of any therapy that uses the body's immune system to target cancer cells. So for a number of years, we've known that the immune system, in addition to just being able to fight infections, it can actually target cancer cells. And there are some experimental results in animals that show that in any given day, there are cancer cells formed in our bodies, but the immune system is able to eliminate them. Unfortunately, that doesn't always work. And in that case, the cancer cells, grow and you find someone with, for example, neuroblastoma. But now we have a number of strategies to redirect the immune system to go after these cancer cells and in the ideal situation to kill those cancer cells. You've heard a lot about the antibody therapy that's been really pioneering this field in children with neuroblastoma and many other cancers. So I won't go into it, but there are other versions of antibody therapies where you can conjugate, for example, a toxic molecule to these antibodies, which when those are picked up by cancer cells, then the cancer can be killed. There are immune checkpoint molecules as well that will not directly target cancer cells, but will kind of take up the brake from the immune cells and have the immune cells attack the cancer cells better. These therapies don't always work for every cancer type, and as Dr. Sondal mentioned, for example, for melanoma, immune checkpoint blockade is a breakthrough therapy, but for neuroblastoma, at least up until now, it hasn't really been showing that level of efficacy. BiTEs are bispecific T cell engagers, again, kind of connecting T cells, which are a special white blood cells, to the cancer cells and have these T cells kill the cancer cells. Many of you are familiar with vaccines for infectious diseases, but now there are vaccines that will redirect the immune cells to cancers. And my talk is going to be focused on adoptive cell therapy, which, there are different types. Even in the subset of immunotherapies, there are different versions. And from the type of adoptive cell therapies that I'll be talking about is with chimeric antigen receptors, or CARs, and we'll get to it in just a second what that means. So how does adoptive cell therapy work? In the current form, we use blood cells to generate cancer-redirected, cancer-specific, kind of fighters. And the blood cells, kind of here are the blood and blood-derived cells that we we all have. Macrophages are kind of derived from blood monocytes. But what I want to point out is this subset of cells. You have your red cells, your granulocytes, various subset of granulocytes, and you have monocytes and lymphocytes. And lymphocytes are the ones that fight infection and as well as able to fight cancer cells as well. There are different types of lymphocytes, and my talk is going to be focused mostly on T cells and to some degree NKT cells. But what I want you to remember is that, out of the number of white blood cell types that we all have in our blood, there is only a small subset of white blood cells that can be used, at least to our best understanding, for genetic engineering to target cancer cells. And the most commonly used type is lymphocyte, T lymphocytes. So how does this work? Well, in the current setting, what happens is that the patient who is diagnosed with cancer goes under some sort of a collection procedure. Sometimes they have to go through a similar procedure when they get stem cells collected, leukapheresis, where a large number of white blood cells are collected. Sometimes it can be done just from a relatively larger volume peripheral blood draw. But what happens is that if ultimately these cells make it to a GMP facility, a manufacturing facility that specializes in growing cells and genetically engineering these cells outside of the human body. This is tightly regulated and controlled, so there are just a number of centers still in the US that can do this. And then once the product is manufactured, after genetic engineering and additional growth outside of the body, in most cases it's frozen, tested, and then later infused back to the patients. This causes some delay. So in most of the cases, this is about a month from the time a patient comes to a center and is donating the blood to the time of the infusion. And obviously during that time, if someone has actively growing cancer, something needs to be done. So most of these patients will receive some bridge therapy until the products are ready. Okay, so chimeric antigen receptors, and these are kind of fusion protein. These are very interesting molecules that are able to redirect these T lymphocytes, these white blood cells, to recognize and then actually kill the cancer cells. What I want to point out here, I don't know if this pointer is working. Oh, there you go. That this chimeric antigen receptor that you see here is actually derived from two molecules. This is an antibody, kind of schematic of an antibody, that you guys are all familiar with from dinutuximab. What's important is that every antibody has an antigen binding site, so an area where that antibody can recognize a target molecule, in our case, on a target molecule in the cancer cell. And you can take that piece, this binder site, and actually fuse it to other molecules. And here I'm showing the T cell receptor complex. So this is a molecule that's responsible for the T cells to be activated under normal circumstances. But obviously this receptor doesn't recognize cancer cells. So how can you overcome this barrier? And what people have recognized a number of years ago, that you can actually fuse this cancer-specific binding part of an antibody to the signaling molecule, the signaling part of this huge complex. And this resulted in the first generation of chimeric antigen receptors. So, and they are expressed on the T cells just like a normal molecule would be. They are crossing the cell membrane. And in the ideal world, this will bind the cancer cell, and this part of the molecule will then trigger the killing machinery, at least of the T cells. Well, now we know that these first generation chimeric antigen receptors are just not enough. You need additional molecules, additional signals to enhance the ability of these CAR T cells to kill, and very importantly, for them to actually survive and expand. Keep in mind that we gave a number of a large number of CAR T cells to patients, but that in the context of a constantly growing tumor, it is a small army, and you want to grow that army inside a patient as much as possible and as long as it's safe. And so with the second and third generation receptors, that became possible. And so these are just molecules here, CD28 and 41BB. Those are parts of these molecules that were added to first generation receptors that make these second and third generation receptors more potent. Whether there's a difference between second and third generation as far as potency, that's still debated, but there is clear evidence now, both in patients and in preclinical models, that the inclusion of any of these molecules in second and third generation receptors is much better than in first. Okay, so this is just a schematic that once you made these receptors that can recognize the cancer cells, after genetic engineering, which usually done to this day by viruses, many times these viruses are actually derived from HIV. These genes are going to be inserted into the lymphocytes, and they actually persist there. So once you engineer a lymphocyte, that gene should be there and would encode the chimeric antigen receptor that then gets shuffled and stays on the cell surface of the T cell. Once that's there, then the T cell can go and find the cancer cells and can recognize these molecules that very often is B7H3 or GD2 or L1CAM. And once it recognizes those molecules, it can kill the cancer cells. But as I mentioned, an important aspect is it's not just killing the cancer cells, but then that these receptors then provide additional signal for the CAR T cells to expand and proliferate so they can continue to kill large number of tumor cells. So do these actually work, you might ask? And we now know very clearly that they work really well in certain hematologic, certain blood cancers. And this was just a number of manuscripts that came out in very high impact scientific journals 10, 15 years ago, showing their incredible efficacy in B-cell leukemias and lymphomas. So that led to the FDA to approve now 6 products, all of them with unpronounceable names. I'm not sure why this happened, but good luck pronouncing them and remembering them. Maybe the brand names are easier. But as you can see, so 4 of them are targeting CD19. Again, this is expressed in a certain type of blood cancers, and BCMA, 2 of them, which is also on a certain type of blood cancers. And here are the diseases that they are targeting. So the big question is, okay, this does work really well, and some of these studies showed up to 90% complete response after a single infusion, and about 50% sustained complete remission at 2 to 3 years out for relapsed refractory leukemia patients. So really remarkable. But can they actually work for solid tumors? And obviously for this audience, can they actually work for neuroblastoma, or against neuroblastoma, I should say? So there have been a number of phase 1 clinical studies conducted around the world, mostly in Europe and the US, and they are very different. So I want to point out that it's very hard to compare results study to study because the patient population they enroll tend to be very different. But nevertheless, they still provide a lot of insight on how they work. And here I just wanted to put up a real schematic of what the genes are that are being put into these cells. And you might think, oh, this is too complicated, but actually not. So here, if you look at this segment, just tells your cell to start reading and start expressing the gene from here. Then you express this thing. This just makes that the expression of the gene lasts longer. And then again, for the second gene, the reading starts from here. You express it, you make sure that it lasts long. And then those 2 molecules, HYTK, and then the chimeric antigen receptor, here with the clone name for the antibody that binds L1CAM, is expressed. This first phase 1 study was conducted by the Seattle Children's Group, and was looking at CD8 positive, so a subset of a white blood cell, T cells that were expressing an L1 CAM specific chimeric antigen receptor and a suicide gene in case just for toxicities. Have you guys heard of L1 CAM as a target antigen for neuroblastoma? Yeah, so it was quite an exciting target about 20 years ago, and I think maybe there's still something to this target, but this study didn't really show good efficacy. And part of it, as I mentioned, was that expansion was limited. So here they treated 6 patients, and from all of these graphs, what you want to point out, what you want to look at is that all these bars show the number of how well these cells are detected in the blood. But as you see the later time points, they all disappear. And this was a first-generation chimeric antigen receptor. So we certainly learned a lot from this study that although outside of patients in the petri dish and in animal models, this thing works. In patients, it didn't really show any exciting efficacy. So with that, a lot of focus went on to try to improve the efficacy. And so at Baylor, a group before my time at Baylor conducted another study with a first-generation CAR, this one actually targeting GD2 and using the binder from the dinutuximab antibody. But in this case, they made 2 products. One was in just regular activated T cells, and the other one was in virus-specific T cells. Those T cells, in addition to expressing the receptor, were also specific for Epstein-Barr virus. Probably 95% of us in the room have been infected by Epstein-Barr virus. We all have this virus integrated in some of our cells. And what happens is that these T cells that now express the chimeric antigen receptor get help from the existing viral infection in our body to persist and survive. At least that was the hypothesis. And they infused 11 patients initially, and then a couple more patients later as part of the NESTLIS-1 and NESTLIS-2 study. When you look at, there were actually some exciting anti-tumor activities. Some of the patients did not have evidence of disease when they came on. They stayed without evidence of disease. Some had, but even some of those who had pretty good amount of disease did have some partial responses. And one of the patients who had a relatively large Calvarian lesion is still alive and doing well. So this suggested that indeed, maybe expansion and persistence is really important. And when you compare the expansion and persistence of CAR T cells, just the ones that are regular T cells, versus the ones that are virus-specific, here marked as CTL, in the same patients, 2 products were infused, and the CTLs were much better persisting longer term. So that led to a number of newer studies, all of them focused on improving the expansion and persistence. And this was a study that I led a number of years ago, but I'll do my best to show you the data reasonably well, where in this case, 3 cohorts were tested. The first cohort was just the cells. The patients came in, 4 patients were infused, and then the safety and efficacy was measured. The next cohort had now lymphodepletion, because there was emerging evidence that lymphodepleting chemotherapy prior to cell therapy actually improves the cell's efficacy. Lymphodepleting chemotherapy is similar to what patients get to target the cancer cells, but in this case, the goal is not to target cancer, rather to eliminate the other white blood cell, other lymphocytes in the patient's body, for lack of a better term, to kind of give room for the infused cells to grow better. And then the third cohort also had the addition of checkpoint inhibitors, pembrolizumab, added after lymphodepletion, and then 3 weeks after the cell infusion, which is marked here as green. So in this case, there is pretty strong evidence that once lymphodepletion was implemented, demonstrated here as the purple group, the expansion and persistence of the CAR T cells was better. And that also corresponded with interleukin-15 concentration in the blood, which is one of the mechanisms how lymphodepletion actually enhances the expansion and persistence of these cells. Nevertheless, unfortunately, the efficacy was still modest. There were patients with growing tumors who got stable disease, but many of the patients progressed through the therapy, unfortunately. So more recently, a British group evaluated a different chimeric antigen receptor. This one was targeting GD2, however, not with the same binder. In this case, they used a different antibody that was fully humanized. One thing is that they used a large spacer. So that is, as I mentioned, there are different components of this receptor, and every component can be changed, and those changes actually do alter the functionality of the receptor. So here the spacer region was actually much larger than, for example, in the trial that I previously shown. And here the costimulatory molecule was CD28. And in addition, they also include a suicide gene just for toxicities. That being said, after a careful dose escalation scheme, when they first gave just the cells, then just cyclophosphamide, then fludarabine, these are the drugs for lymphodepletion. And after that, they started to dose escalate all the way up to 10 to the 9th cell per meter square. They did show some better expansion and persistence in the higher dose levels. And they were able to show some objective responses. Here's a planar imaging of MIBG scan, where, for example, if you look at the vertebrae, there is certainly a decrease in MIBG avidity. But unfortunately, there weren't any durable objective responses in this study. So, okay, now the persistence can be improved, but the efficacy is still not there where we wanted. And I think one of the exciting results came just recently from an Italian group that used a third-generation chimeric antigen receptor targeting GD2. The binder is from the dinutuximab antibody. It actually has a short spacer. It has both the CD28 and 4-1BB costimulatory domain. And it is by far the largest chimeric antigen receptor T-cell study to date. And in this case, 3 patients were infused on the first dose level, 3 additional ones on the second dose level, and then 21 on the highest dose level. Here the dosing is kilogram base. So this would be roughly 3 times 10 to the 8 CAR T cells per meter square. And then they did show quite good long-term persistence. So the peak was reasonably good, but then you can detect the cells all the way out close to 2 to 3 years now at a reasonable level. It is not as high as in the leukemia patients, but they are detectable. And what was interesting is that they certainly showed some promising results. This is a swimmer's plot, and let me just walk you through. So on the top level, you see that the color scheme is corresponding to the response itself. So orange is the one that you want to look at that's corresponding to complete response. Purple is partial, stable disease is blue. And you see a number of complete responses there, which is certainly encouraging. I want to highlight, though, that some of these patients came in without evidence of MIBG-avid disease. So it is difficult to know, whether those patients really had active disease or not. And same thing, some of the patients did have MIBG-avid disease, but after therapy, they had partial response that was durable, in this patient's case, up to 3 years, which, we are not sure whether these patients received additional therapies. It was not mentioned in the manuscript, or whether this was already MIBG-avid, but more of a mature kind of ganglioneural disease. Nevertheless, this is really promising because there are some encouraging responses, even in patients that have MIBG-avid active disease. And this is just one example that they included in the manuscript. Here you see this massive tumor, that's MIBG avid, that shrinks nicely after infusion. So certainly there is some movement in the field, and I just wanted to quickly show one of the studies that our group runs. In this case now, as opposed to T cells, natural killer T cells are tested. The reason for this is because these NKT cells can effectively infiltrate neuroblastomas and target cancer-supporting tumor-associated macrophages. So another white blood cell type in the tumor that we know is actually bad for the patient. It helps the tumor to grow. And in this case, it's a second-generation receptor that used. The binder is from the GD2 antibody. And in addition, these cells produce IL-15. As I mentioned, IL-15 is one of the mechanisms how lymphodepletion helps the cells to grow. So in this case, the cells actually produce some of it. Well, we haven't talked about toxicities on the context of CAR T-cell therapies, but in general, up until now, the safety of these therapies have actually been really encouraging. And I just wanted to show the safety table for one study because many of the others have basically comparable results. And what you can see is here that the grade 3 and 4 toxicities, so the higher the grade, the more severe, are all dominated by anemia, low white blood cells, low platelet counts. And this is due to the lymphodepleting chemotherapy that these patients get. So this is expected. This is not product related. And one other encouraging aspect is that even in the context of targeting GD2, there isn't really detectable pain-related side effects. You see some here, but these were reported, but actually these were coming from patients who had progressive disease and were usually related to involved sites that became painful. In this study, there were some objective responses again. Here, the color coding is different, just to confuse everyone. So here you want to look for green and yellow. Those demonstrate anti-tumor activity. And here is an MIBG, and this is the planar scan, and here are the specs, where the red circles show the involved disease and how that disappears after the infusion. Keep in mind that the cardiac muscle can take up MIBG, so this is not disease, but this part is, and so is this area. So that being said, well, if you are interested, and I hope that this short presentation gave you enough insight and perhaps triggered some excitement toward these trials, because I do believe that these can hopefully change the way we treat cancer and particularly neuroblastoma moving forward. So I wanted to highlight, to my best knowledge, the current investigations in Europe and in the US. And just going over first, this study is testing, evaluating glypican-2 specific CAR T cells. So this is a novel antigen that that was identified by John Maris's group at CHOP, and now there is a phase 1 clinical study evaluating the efficacy of GPC targeting CAR T cells. Another trial that is still ongoing targeting GD2 using a second generation receptor in T cells that also co-express IL-15 is running at UNC, and I put up actually the contacts here for you all. A third study that you might want to consider is the one that I just mentioned and showed the results for, is the GD2 CAR NKT cell study that's run at Texas Children's, and it is targeting GD2 with the IL-15 and GD2 CAR expressing NKT cells, and Gangwen Tian is running now the study. The trial that I mentioned from the Italian group is now in the phase 2 setting, so they are looking at efficacy, not just safety primarily. It continues to target the GD2, and Franco Locatelli's group is running the trial still in Rome. Another trial that is recently opened at St. Jude's Children's Research Hospital is targeting B7-H3. This trial is run by Chris Durenzo primarily, And this is exciting as similar to GPC2 is looking at another target antigen. Bilal Omer's group at Baylor at Texas Children's is also targeting GD2, but in this case, they are trying to improve the efficacy and anti-tumor activity and persistence by using a new molecule that's constitutively giving interleukin-7 signaling to the cells to survive better. And then lastly, Crystal Makal's lab developed an approach to target GD2 with a second-generation CAR that's expressed in T cells, and they use a unique manufacturing procedure that enables the cells to be less exhausted while they are being grown outside of the body, and this study is now running in a multi-institutional setting. So I hope that these might be interest of you, and I hope that if you are in the situation when some of these might be relevant to you, then feel free to take a picture and spread the word. So that being said, I think thus far we've demonstrated, the field has demonstrated that CAR T cells are safe in children with neuroblastoma. The persistence and expansion seems to be a really critical aspect of anti-tumor activity. Complete remissions now can be achieved in children with neuroblastoma. Some of them can be perhaps even durable. And then there are several studies. It's really, really exciting, and I think we should be very optimistic that some of these studies are going to be finding finally strategies to induce durable complete remission in children. And with that, I just want to thank all the patients, families, and referring physicians who really get these studies enroll efficiently. And this is my lab. Obviously, I benefit a lot from them. And Texas Children's has a neuroblastoma program, and with the leadership of Dr. Foster is really an exciting group to work with. And those are my contacts if you need to get in touch. Thank you.
Does anyone have questions?
Thank you, Doctor. I have two questions. One, is CRS a concern when talking about CAR T cell therapy and neuroblastoma? And two, has there been any translational research done specifically in regards to machine learning to try to predict efficacy and toxicity for patients going through CAR T cell therapy for neuroblastoma, specifically seeing if it can be done in an outpatient setting?
Yeah. So the first question, CRS, cytokine release syndrome, thank you for bringing it up. That's indeed very much a concern still. But nevertheless, I think so the cytokine release syndrome is well described in patients with leukemias and so they're blood cancers and occurs in the majority of patients and is actually associated with good anti-tumor activity. I think, we learned so much about CRS now that we understand how it's mediated with interleukin-1 and interleukin-6 signaling, and there are specific drugs that can disrupt it. So I think unless there's a different mechanism for cytokine release syndrome in the context of neuroblastoma, I would not be necessarily too concerned about it. I'm not saying this is not an important safety aspect, but I think it would be very unlikely that we infuse CAR T cells, nothing happens and the tumor shrinks. I think if these cells get activated, just like when we get a fever, when we have the flu, and these cells are activated and fighting the virus, we should expect some of those febrile illnesses in patients where the cells are working. Of course, we don't want it to go out of control. So, how to mediate this is certainly going to be an important thing to look at. As far as, AI and machine learning and all those, I think that's going to be really exciting to see to evolve. Right now, there's just so few patients, right, that it's very hard to train a machine when you have, an N of 12 to input. But later on, certainly, yes, because the number of analytes that we can measure now is just so many that it will be certainly helpful. I hope so, yeah.
Anyone else? Yep, go ahead.
So yeah, to kind of piggyback a little bit on the CRS, you explained there's a little bit of there's like medication, steroids, or whatever that can cut down on that inflammatory response. Are there any other, like would you give like chemotherapy to maybe help that as well? Or I guess, yeah, like are there other ways to help a CRS response calm down
Yeah.
to preserve a life, like, that kind of thing?
So there are a number of drugs out there that can be used to temper CRS if it happens. Tocilizumab and anakinra are the primary ones that people look at, targeting IL-6 and IL-1. Those are two molecules that mediate this primarily. And then if that fails, there are a number of other strategies that can be employed. So tumor necrosis factor alpha inhibition is one of them. Then, giving steroids. And ultimately, one can just give lymphodepletion again and to eliminate the cells. That being said, that's what everyone is trying to avoid, because once we go past the direct immunomodulation of, TNF-alpha or interleukin-6 inhibition, we are going to hit the tumor-specific cells as well. And maybe if they are overactivated, it is fine to temper them. But once we get to like chemotherapy, let's say someone gives fludarabine to someone who has CRS, I think that would probably eliminate the anti-tumor activity as well.
Okay.
And then that just, there are some attractive strategies to include, safety switches in these CAR T cells that can ideally dial down, not just turn off the cells, but dial down their efficacy. So for example, the Italian group did have the inducible caspase-9 in their construct. It was only used once in the case of a central nervous system bleed, which was actually not even related to the study. But there are now genetic engineering strategies to help with this. Yeah.
Okay. And sorry, I kind of have like two more to build upon.
Yeah, sure.
In your experience with like CRS and disease load, like is there any correlation with how, minor or severe a CRS response would be in regards to disease load, like low versus high disease load?
So I think from neuroblastoma, we don't have strong correlation yet because there's just very few patients who've been treated, and then, of course, very few patients actually had CRS. But in the leukemia world, higher disease burden corresponds with higher risk of CRS.
Okay. And then, sorry, last question. With some of these slides you shared, like there's like a partial response, right? So obviously there's still disease present in the body. You want to still preserve those CAR T cells, right? That have helped, that are doing their job. Are there therapies that should be avoided down the line to help preserve those CAR T cells so that they can still do their job while we're still working on, knocking down more disease?
Yeah. So before I get there, actually, one thing to consider is that these infusions can be done multiple times. So that's something that I think being evaluated, whether additional infusions are helping at the same dose. And then another interesting aspect is whether one should be able to do safely intrapatient dose escalation. In other words, if someone was infused on dose level 3 and has a partial response and had no major toxicities, can we actually dose escalate to dose level 4 in the same patient and see if now the higher cell dose will eliminate the disease while maintaining safety? So those are all questions that I think the field will try to answer in the next couple of years, hopefully. And then going to the other aspect that you mentioned, what should we avoid? So in general, after CAR T cells, if the patients go back to conventional chemotherapy or high-dose MIBG therapy, that is going to be toxic to the CAR T cells. So those will probably eliminate the cells. That being said, are there other immunomodulatory therapies that could help? For example, PD-1 inhibitor, checkpoint inhibitors. Would they be helpful? I don't know. We did try in 3 patients, and they didn't make a difference. But again, it's 3 patients. And there are newer and newer checkpoint inhibitors. So there might be something out there. I think that will have to be carefully assessed in the context of a clinical study.
Okay, thank you.
Anyone else? Any other questions? Nope. Okay, thank you, Dr. Heczey. We're actually on time. This is crazy. Anyway, we're going to take a 15-minute break. We have some snacks and stuff in the other room, so come back at 3:15.